So let's start. Okay, so we are going to do first the nucleic acids. Okay, we are going to do first nucleic acids. So this is very easy. See, nucleic acids. All right, now what is nucleic acids? The name itself tells you. Nucleic means this is something related to nucleus. They were discovered in nucleus. And acid because they have acidic nature. They have acidic nature. So basically, where do they occur? They occur in a cell within the nucleus. If this is the nucleus, this is where we find the DNA or nucleic acid. Okay, in higher. In terms of bacteria, we will find it in find it in a specific region which is called as a nucleoid. Okay, so now very important, what is the important function of nucleic acids? Nucleic acids, when they were first discovered, okay, they were called as nuclein, but later they gave the name nucleic acids because they found acidic character, they found acidic nature in these structures. Okay, then next, what is the most important function which attracted the study of nucleic acid? The important function is that these nucleic acids, they are nothing but they store genetic they store genetic information. They are the storehouse of genetic information. Whatever information related to our genes, heredity, which is required to pass on to the next generation, any information pertaining or information related to our body. Okay, the straight hair, the color of the eye, the color of the skin, anything, everything is controlled by the nucleic acids. Everything, all information is stored in the nucleic acids. Okay, so they store the genetic information. Whatever is there in our body, our skin color, okay, our height, okay, our hair, any function that is going on in our body, if somebody is suffering from any diseases, anything, every information is stored in the nucleic acids. And how it appears, for example, let us say, okay, suppose for example, we all have a particular cell. This cell is called as proto-oncogene. Okay, we all have, and this protects our body from any kind of cancerous growth. All right. Now, if it is so, then all people of the world should suffer from cancer, but it does not happen. There's only selected people or only few people who suffer from back cancer. Why? Because that is also related to the gene. Every one of us, in some people who suffer from who suffer from cancer, their genes are altered. That proto-oncogene becomes oncogene and it causes cancer. But it doesn't happen in everybody. So that is also, that is there, that information is stored in the genetic material, that is the nucleic acids. Whatever function takes place in the body, doesn't matter what it is. Production of hemoglobin, how much hemoglobin is produced in your body, how much is produced in my body, that is also stored in our genetic material. Okay, in fact, whatever is happening in our life, every bit of information is stored in the genetic material. This is one of the most important point that you need to know. Okay, they store genetic information. Not only that, they store, first of all, they store, and whatever is there, they help forward in carry forward of genetic information, carry forward of genetic information from one generation, from one generation to the other. This is important. Okay, from one generation to the other. Like we get some characters of our parents, but we don't look alike exactly like them. But we have some features, all right? That is also there in the genetic material, all right? So everything which is there in us, or everything which happens in us, every information is stored in our genetic material. Now, for example, let us say the people found fossils of dinosaurs. Okay, initially they found fossils of dinosaurs, but later on, how even we came to know they look like, how they look like. We just had the skeleton, right? We just had the skeleton. We just had the fossil, and not even say skeleton. We just had the fossil. How they looked, what was the condition of the earth at that time, what they ate. Okay, what was T-Rex, or what was the other dinosaurs. How did we know? We did their DNA analysis. Whatever, every information was stored in the DNA. That is why genetic, the nucleic acids are so important. When we talk about the nucleic acids, okay, when we say about the nucleic acids are of two types. Okay, the nucleic acids are of two types. Now we'll see. The first one is called as the DNA. The next one is called as the RNA. Okay, DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. This is the genetic material. However, DNA is one of the most common genetic material. Common genetic material. Okay, common genetic material in all organisms. RNA would be the common genetic material in viruses, but that does not mean that RNA is not found in us. Yes, it is found in us. Yes, it is found in us. Okay, but RNA plays a different role altogether. Okay, so till here, is it clear?
Yeah.
Okay, now let's try to understand these two genetic materials. Okay, so when we talk about genetic, when we talk about the nucleic acids, so if I say nucleic acids, nucleic acids are made up of what? Nucleotides. This is the word that we use, whether it is DNA or RNA. They are made up of what? Nucleotides. Okay, so what does nucleotide have? It has three substance, okay, three substance combined, three substance combined together, three substance combined together. What are they? The first is, okay, the first is, let us see. All right, let me see. Yeah, okay. The phosphoric acid. Phosphoric acid. This phosphoric acid it gives the acidic nature to DNA. Because of the presence of the phosphoric acid, our DNA is acidic. That is why we say deoxyribonucleic acid or ribonucleic acid. Whether RNA or DNA, they contain phosphoric acid, and this phosphoric acid is responsible for the acidic character or for the acidic nature. The next is the pentose sugar. Pentose sugar. Okay, pentose. 5 carbon atoms are there, so it is called as pentose sugar. And In DNA, the sugar is deoxyribose sugar. Okay. In RNA, it is called as ribose sugar. Okay. To
Mm-hmm.
be very precise, let us even look into the structure of the phosphoric acid. This is how the phosphoric acid structure will look like. The OH bond, we have hydrogen and a double oxygen. This is how the phosphoric acid looks like. When you get this structure, immediately know it is the phosphoric acid structure. Let us see next the structure of the deoxyribose and the ribose sugar. The deoxyribose, okay, we have, now see here. Okay, so this is a ring structure. Every corner of the ring is for a carbon atom. This is one, this is one, this is one. And we have a carbon atom which is also outside the ring. Outside the ring. Okay, outside the ring. Now let us arrange this. Okay, so we have, let's see, we have a hydroxyl or OH group, H. This would be H and H. And then again we have an H and OH. Let's see. Then H and here, okay, we will have CH2OH. So this C is the C only, nothing. CH2OH. So in total, how many carbon atom?
5.
One, two, three, four, five. Right?
Yeah.
Yeah. So it is called as a pentose sugar. So it is called as a pentose sugar. Observe this position, carbon 2 position. Okay, we'll see. Next, ribose sugar. Ribose sugar also have a similar structure. Ribose sugar also has a similar structure. Let us see here. So 1, 2, 3, 4, and then we have here the fifth one. So let's see here. All right. So we have OH, H, H, and OH, H, OH. Oh, sorry. Okay, OH, H, and CH2OH. You observe the carbon 2 sugar, see? And one more thing, here also 5 carbon. 1, 2, 3, 4, 5. Pentose sugar. Observe carbon 2. Observe this and observe this. This is the major difference in the deoxyribose sugar. In position of carbon 2, we have a hydrogen. In ribose sugar, we have a OH group. Deoxyribose sugar, this is in DNA. Ribose sugar, this is in RNA. This point is clear, Sana? Have you
Yeah.
understood this? Yes.
Mm-hmm.
Okay. Keep this in mind. Okay. All right. Then, now, after carbon sugar, now you have to understand that the phosphate and the sugar, both of them make up the backbone. Again, I'm repeating, backbone of the DNA molecule. Very important. These two together make up the backbone of DNA or RNA molecule. Okay, so when we say backbone, very for mostly DNA. Next is the third one. We have our nitrogen bases. Nitrogen bases are of two types. One is called as the purine nitrogen base, and another one is the pyrimidine nitrogen base. The purine are larger compounds. Purine are larger compounds, and they have double ring structure. See, one ring is this, the second ring is this. Pyrimidine are smaller nitrogen bases. They have a single ring structure, only one. This is the difference. Next, we will also have, okay, here the two type of purine, adenine and guanine. This is denoted by A, this is
Mm-hmm.
denoted by G. Pyrimidine is thymine, cytosine, and uracil. Thymine is T, cytosine C, and uracil is U.
Mm-hmm.
So, out of this, adenine is present in both DNA and RNA. Guanine also is present in DNA and RNA. Thymine exclusively in DNA. Cytosine, DNA and RNA. Uracil, it is RNA. Okay, uracil is what? RNA. This point is clear?
Mm-hmm.
Okay, so structures you need to know. Then, for example, Sana, if you remove the phosphate, see, three compounds are there. Okay, three compounds. One is what?
Uh,
Phosphate,
so
sugar, right? And
and
what is the third one?
nitrogen base.
Nitrogen base. That makes a nucleotide.
Mm-hmm.
Remove the phosphate from the nucleotide, you get a nucleoside. Clear?
Mm-hmm.
Okay, this is important because they may ask you question on, like, the nucleotide and nucleoside. This distinction is very important. Okay, so you keep in mind what is nucleoside and what is nucleotide. Now, what happened? We have scientists like, Rosalind Franklin was there, Morris Wilkins were there. The names are important.
Mm-hmm.
Rosalind Franklin, what she did was she studied a DNA and she took the X-ray crystallographic image. Okay, X-ray crystallographic image. You can see this image. Can you see this?
Yeah.
Yes, X-ray crystallographic image of DNA. All right, so she studied this DNA and she took the picture. That was the first picture ever anyone has taken on DNA. First picture.
Mm-hmm.
And nothing more was known about this. Morris Wilkins also worked on that. Rosalind Franklin and Morris Wilkins, they produced X-ray diffraction patterns, okay, on of DNA. So both of them. But more research was there. Then came Watson and Crick. Okay, Watson and Crick, what they did was, in they after years of their research, okay, after years of their research, what they did was they used the picture which Rosalind Franklin and Morris Wilkins had given. They used it and they made a model. Okay, and they made a model. If you can observe this, see, they made a model. Can you see?
Yeah.
Yeah, this is important, Sana. They did not produce, they made the model. That's why we say double helical model of DNA. They
Okay.
use the information of Morris Wilkins and Rosalind Franklin, and also they gave what Erwin Shergaff has given. So what they did was they did use a model, double helical structure of model. This was like, it was like a storm. It was like a tsunami in, molecular biology. Because after this, there were so many things that came up. Watson and Crick, Rosalind Franklin, Morris Wilkins, all of them received Nobel Prize for their immense work on DNA.
Mhm.
Okay, all right. So we'll study now the structure, double helical structure of DNA, which was given by Watson and Crick, or we also call as the double helical model of DNA. This was given by whom? It was given by Watson and Crick. So let's just start. We try to understand this and everything will be just clear, crystal clear. Just I'm going stepwise. You just listen very attentively. If you get stuck in between, please tell me. I'll repeat that again. Clear?
Mhm.
Double helical. What I mean by double helical is take a ribbon. If you take a ribbon and you just roll it over like this, okay, you get a helical structure. All right, you get a helical structure, okay, folded structure. That is called as helical, and that's how our DNA is there. Okay, that's how our DNA is there. So DNA has a double helical structure. So our sugar, this is where the sugar will be located. Here will be the phosphate attached to the sugar. This is the phosphate, and between this, everything between this, between the two strands, is nothing but the bond that will keep the two nitrogen base together. This is a bond. Okay, it's a bond that will keep the two nitrogen base together. This is how the DNA will look like. Okay, to
Mhm.
understand it more simplified manner, let's see. First of all, the DNA, our DNA is helically coiled, helically coiled, and we see two strand here. So helically coiled, it is double stranded. Okay, Watson and Crick model, DNA structure, is it there for the exam?
I think so.
DNA is there, right?
Yeah.
Then I'm sure it should be there. Okay. All right. We continue. Okay. Double helical DNA. Okay. So double stranded. Two strands are there. All right. Okay. So DNA is double stranded. RNA is single stranded. So we have double stranded DNA. Now, suppose if I open this, okay, if I open this, this is what I will get. Two strands of DNA. All right. And these strands of DNA, we say it, they are anti-parallel. They're anti-parallel. That means they're running in opposite direction. If we call this is 5 prime, then this will be 3 prime. Opposite, this is 5 prime, this is 3 prime. Anti-parallel. Okay. Then the diameter is 20 angstrom. Diameter diameter of DNA is 20 angstrom. When when you roll the DNA, you get two grooves. This is termed as minor groove. This would be termed as major groove. Okay.
Mhm.
Now, DNA is turned one helix, one turn, one turn, or one helix, or we also say a single pitch. One turn, one helix, or single pitch contains 10 base pair. Base pair means nitrogen bases. 10 base pair, 10 base pair.
Mhm.
Okay, we'll see what is this base pair. 10 base pair. And So the measurement, one turn measures 34 angstrom, okay, and one helix 10 base pairs. Now, Sana, you please also keep in mind that when we are doing this DNA structure, we what we do is that not only in angstrom, in nanometer also we measure this. Okay, nanometer also we measure. So sometimes you may get this as 3.4 nanometers. So don't get confused. Okay, in nanometer also we measure, in angstrom also we measure. Okay, 3.4 nanometer. Sorry, 34 angstrom. Next, let's see here. This is one nucleotide. This is another nucleotide. They're joined. Nucleotide means phosphate, sugar, nitrogen base join. So the distance between these two, the distance between these two, okay, I can say distance between, okay, two nucleotides, okay, is 3.4 angstrom.
Mhm.
Distance between two nucleotides is 3.4 angstrom. Okay, now let us see this. This you try to understand what I am drawing now. Okay, Sana? Be
Yeah.
attentive now. Okay, see this. We have one strand of DNA, and let us go to another strand of DNA. Clear? Two strands of DNA. Okay. So now we will draw now a sugar, deoxyribose sugar. Clear?
Mhm.
Okay. Can you see? See
Yeah.
how they are placed? See how they are placed?
Yeah.
Okay. Now, when you do this for the first time, it's very difficult. So what I advise you to do, I'm advising you do this. Take a notebook. In that notebook, you write it down first. Okay. You write down whatever that this picture you draw it. Then what you do? Take your notebook upside down and draw the other strand. Okay. Now see this very nicely because it's anti-parallel, right? It's anti-parallel. It's running in opposite direction. So we have to draw this sugar also in opposite. Can you see this?
Yeah.
See, right?
Mhm.
See, this is how you draw. Clear? So we have two strands. Now, let us see here. Okay. DNA ribose sugar. So now can you tell me the number here? I'll give a blue color dot and you tell me which number carbon atom it is. Okay. This one. What is which number carbon atom?
5.
5. This
3.
3. Remember this 5 and 3. Remember this 5 and 3. Okay. Now, what I want you to do is that if you have a notebook in front of you,
Yeah.
stationery is there. Yes. Draw the structure. Draw.
Already.
You have drawn?
Mhm.
Yes. Now, when I'm explaining you DNA, you have to refer to the structure. Okay. All right. So now you see here very nicely. This is one strand. So this is 5 carbon. Clear?
Mhm.
This is 3. What is this? 5, 3, 5 again, 3, again 5, and again 3. So what do we have at the top?
5.
5 prime. 5 carbon atom. So
Mhm.
this becomes 5 prime. What do we have at the bottom here?
3.
3. So this becomes 3 prime. Will this be same?
No.
Yes, but is it opposite, right?
Yeah.
So what will be here?
5.
5. 5 prime. And here?
3.
3 prime. Did you get that?
Yeah.
Yes. Okay. Now, I'll erase this. Okay. Now you see. All right. See now. Okay. Now you see. We have a phosphate here. What do we have? Phosphate. We have another phosphate here. Then we have a phosphate. Each between we have a phosphate. See? Now, see here. This phosphate is attached here, the one in between. Here, if a phosphate is there, it will be attached to the 5 prime, right?
Yeah.
This phosphate is attached to the 3 prime of the first sugar, and this is attached to the 5 prime of the second sugar. This is clear?
Yeah.
Similarly, here also 5 and 3, like this. So, what is phosphate doing? Phosphate is holding the sugar, right?
Yeah.
Yes. Did you understand this, Sana? Any doubt?
Yeah. I understand.
Yes, clear?
Mhm.
Okay. Now, so phosphate and sugar are called as backbone. Now the question is, where is the nitrogen base to complete the DNA? Let me take from here. One and straight. One nitrogen base. Okay. Then to bond, one and straight. One nitrogen base. Did you get my point now?
Yeah.
Yes. Now suppose, for example, okay, according to Erwin Shagaf, okay, he said a purine will always bind or make a bond with pyrimidine and vice versa. He also said that adenine will bind with whom? Thymine. By, can you see this? Two, I've given two straight lines, right?
Mhm.
That means two hydrogen bonds. Side guanine will bond with cytosine. Can you see how many straight lines I've given?
3.
Three. So three hydrogen bonds. Okay. So suppose, Sana, for example, if here is adenine, what will be here?
Thymine.
Thymine. How many bonds?
2.
Two. Again, if we have a guanine here, no, we'll not have a guanine because it's purine. So we'll have a cytosine here. So what will be here?
Guanine.
Guanine. How many bonds?
3.
Three. See, this is how they are arranged. Can you see that?
Yeah.
Yes. Okay, Sana. From this diagram, you tell me, where is the nucleotide? Where is the nucleotide?
The nucleotide.
Yes. This
Um.
one. This one. Sorry, sorry. One second. Okay. This one. Phosphate, yes.
Mhm.
Sugar, rather than that, I'll not draw this, one second. Yeah. See this. Phosphate, sugar,
Nitrogen bases.
yes. These three dots that I highlighted, these three regions that I've highlighted, this makes one nucleotide. How many nucleotide?
1.
One. So in DNA, do we have one nucleotide? Do we have one nucleotide or many?
Many.
Many. That is why DNA is called as polynucleotide chain. Clear?
Mhm.
It is called as polynucleotide chain. Did you understand?
Yeah.
Till here, is it clear?
Yeah.
Yes? Okay. Now we have to see DNA replication. Before we start DNA replication, you need to clearly understand one very important fact. Okay, this was given by Crick. Crick gave an explanation. We call it as the, sorry, central dogma of molecular biology. Okay, central dogma of molecular, 1 second, 1 minute, biology. Central dogma. According to him, the RNA, sorry, according to him, the DNA, which is our genetic material, this is able to self-replicate. What do you understand by the word replica?
Like clone itself,
Replica. What is replica?
like make the same.
Same. Form same of its kind. A clone. So DNA is able to form its own replica. And he named this process as replication. What he named?
Replication.
Replication. He named it as replication. So we call it as what? DNA replication. Clear?
Yeah.
This point is clear?
Yeah.
Okay. All right. Okay, so replication. All right. Now, from DNA, we can form RNA. Okay, from DNA, we are able to form what? RNA. That means whatever the genetic information is stored in the DNA, we are passing that to whom?
RNA.
RNA. Because we want to express that. Suppose, for example, I need to have a skin color. That information is there in DNA. That information is there in DNA. Okay, that information is there in DNA. But unless and until my genes are expressing that character, I will not have a skin color. Did you understand this, Anna?
Yeah.
Yeah. So DNA cannot express itself. So it tells, okay, listen, I cannot express. I have so much of information. I don't know how to express. RNA, I'll give you this information. Please do something for me. RNA says, okay, you give me. So what happens? DNA will give the information to RNA. And from DNA, we get what? RNA. Okay, the process of making of RNA from DNA. This is called as what? This is called as transcription. Sana, see the word. Very easy. Molecular biology is very easy. You just need to know. Trans. Okay. And scription. Script. Trans. Translate. Script. The script or the scripture. DNA has information able to express. So what it will do, it will translate its information, and that translation it will keep in DNA. So the process of making of RNA from DNA is called as what? Transcription. Sana, this is understood or not?
Yeah.
Yeah, clear?
Yeah.
Transcription. Okay, next. RNA has all the information now. RNA has all information. DNA has translated. DNA's language? DNA's language is called as code. And that code is translated. It's translated in some other language and put in DNA. And we call it as codon. Now, RNA has so much of codon, that is information. Everything in DNA is translated to RNA. RNA says, okay, now I will do the work of expressing. So, the last phase of expression is where RNA will help in the process to make what? Protein. We call as what? Translation. Translation, or we call it as protein synthesis. Translation, or what we call it as protein synthesis. Clear?
Yeah.
Okay. All right. Okay. Till here is it clear? So whatever is DNA, now it is made into protein. Now this protein will show the different characters. Okay. So you study this in various phases. First you study replication, then you study transcription, then you study translation. So what we are going to study now is the process of replication. Is this point clear?
Yeah.
Yes, till here. Any doubt, Sana? Any doubt?
No.
Okay, great. All right, okay, fine. Now we will see the process of we'll see the process of what? Replication. Okay, we'll see how the replication takes place, how the formation of new genes takes, formation of RNA, or how the DNA will be formed. So let's start. Okay, all right. Now, so replication. So see, Sana, here we have our highly coiled DNA, right? Hmm. So what we need to do is, yeah, we cannot replicate in this way. So okay, replication will take place inside the nucleus. Where it takes place?
Nucleus.
Nucleus. Because our DNA is present inside where?
Oh,
Nucleus.
nucleus.
Yes, okay. Now see this. So this is the DNA. So there might be a point in the DNA. I'll just put a dot here. There might be a point in the DNA, and in this point, okay, this point is referred to as the ori site, or site of origin of replication. That means it is this point, this part of DNA where replication will what? Start. Okay, replication start. So to start the replication, you have to separate the strands. Okay, the strands are together. You have to separate them. So Sana, can you tell me what bonds have to be broken to separate the strand?
The
What was that bond between the two strands of DNA?
hydrogen bonds.
Hydrogen bond. You have to separate, you have to break the hydrogen bond if you want to separate it. Okay, till here is it clear?
Yeah.
Okay, so here we will have an enzyme. We'll have an enzyme. Let us see here. Okay, yeah. We'll have an enzyme that will come and attach at this region. Okay, and this enzyme will break the hydrogen bond. This is called as helicase. Helicase will break hydrogen bonds. And this is how we will get. See here. Okay. See? Totally separated. Okay. See, this is hydrogen bonds are what? Exposed. All right. Whereas, see, can you see this, Sana? Yes.
Yeah.
Yeah. Okay. So from both side replication will take place. So we have a helicase attached on this side. We have a helicase attached on this side. So ultimately, only one side I'm going to teach you. Okay, but I'm just showing you the side. This is clear?
Yeah.
Now, Sana, very important. Our DNA, no, it is very, it's like, it is so much attached. Sometimes we are attached to our friends, right? You have a friend in school?
Yeah.
Yes, your best friend. Yes or no? Yes, that single friend with whom you share everything and you share your lunch and you go out together, right?
Yeah.
Yes, okay. Yes, and the single friend with whom you fight, if you fight, you will not talk for days. Am I right?
Yeah.
Yes, correct. Correct. Very correct. So DNA, the two strands of DNA are best friends. They always remain together. But now helicase have broken the bond, have separated them. They are so much attached, they are so best friends that they tend to come to close to each other. And again, the DNA unwind. So if DNA unwinds, we cannot carry out replication, right?
Yeah.
So we have to maintain that single stranded. So there are some kind of proteins, these proteins will attach on this strands, and they will not allow the two best friends to come close to each other. And we call this as SSBs, or single stranded binding protein. Clear?
Yeah.
Okay, so now two strands are separated together. Okay, so what we will do next, Sana, we will take only one part now and we will concentrate in that part. Clear? Okay. So now, can you observe something? Because helicase has opened the strand. So here, what happens, Sana, if you take a rubber band? You take a rubber band, and what happens, you try to suppose this is the rubber band, and if you coil it in one section, and if it's highly coiled, suppose you take a thread, that will be best example. You take a thread and you open the strands of the thread. What happens? What you observe, when you take a thread, suppose threads, okay, they're like this, right? And when you separate this, what happens? When you separate this, isn't there a stress generated at this region? This region will be highly coiled, right? And this is already separated like this. Yes or no? Yes. Okay. So same here, when you are separating the DNA, see, when you're separating the DNA, this is separated, this region is separated, fully separated. But what about this region behind the helicase? It is under stress. This region is undergoing what? Supercoiling. Okay, so we have enzymes that will release this stress. Okay, so we have topoisomerase or gyrase. This will relieve the stress in supercoiling. I'll write here. Relieve stress in supercoiling. This point is clear?
Yeah.
Yes? Okay. Now let's see the diagram below. Okay. So now one more thing before that, before we start. Do you see something like a Y-shaped structure? Do you see a Y-shaped structure? See?
Yeah.
Yes. Because the DNA strands are unwind, they form a Y-shaped structure in both sides. This Y-shaped structure is called as replication or replicating fork. What is it called? Replication or replicating fork. Now we will take only one side. Sana, are you understanding?
Yeah.
Is it confusing?
No.
No, clear? Okay. Let's see now. In school, have they taught you about these enzymes?
Yes.
Yes, right? This school, you just have to remember the names. Okay, that too, I'm not giving you the prokaryotes and the eukaryotes there. Okay. All right. Okay. So when we do this chapter in detail, all right, there are some more points right now because your exams are there. So I'm just revising this chapter. Clear?
Yeah.
Okay. So replication fork. Let us see. This is 3 prime. So this will be what? 5 prime. So this is in turn 5 prime. This is 3 prime. So this is the replication fork. This is the replication fork. Okay. So now see, the two strands are separated. Sana, if suppose, for example, the two strands are separated. Now, how will you make a new strand? Can you tell me? How will you make a new strand?
You replicate it?
Sorry?
I'm not sure. You replicate it?
Yes, we will replicate. But see, this is what is this? Isn't this the old strand?
Yeah.
Old or the parental strand?
Mhm.
Old or the parental strand?
Yeah.
We call it as what strand? This is called as a template. What strand? Template strand. Clear?
Yeah.
Next. So what we are going to do, we are going to make a new strand here. Okay. This is the new strand. Later, this is going to cut, and then we are going to have a new DNA. A DNA which will have an old strand and a new strand. So this is our DNA, new DNA, replicated DNA. Clear?
Yeah.
Right? Okay. So let's see the structure. Replication fork. This is important. You know, you get many questions from this replicating fork, replication. All right. Okay. So let's see here if this is 3 prime. So now please remember the 3 prime and the 5 prime.
Mhm.
Okay. 5 prime, 3 prime. I'm making it a little bit easy, the diagram, so that you understand. Okay. Now, what was here? We had an enzyme. Okay, we had an enzyme, and this enzyme is called, it was called as which enzyme? I'm giving the same color. Can you tell me which enzyme? Which separated the two strands?
The
Helicase. Yes or no?
helicates.
Yes. Helicase enzyme. So right here. This is helicase. Here we have the strands. Okay. So now you see the process of replication. Always remember the process of replication will take place from 5 prime to 3 prime. From where to where? 5 prime to 3 prime. Helicase enzyme. Okay, replication. This is very important. Very important. Replication proceeds from 5 prime to 3 prime. This is okay. So let us see here 5 prime to 3 prime. That means the old strand. See here, Sana, here the old strand. Can you see this? Isn't this the template strand or the old strand? Template strand, right? Or the old strand? Yes or no?
Yes.
Yes. This is in which direction? 3 prime to which direction it is?
3 prime to 5 prime.
5 prime. So if I am forming a new strand, this will be in which direction?
5 prime to 3 prime.
This will be 5 prime, and here it will be 3 prime, right?
Yeah.
What about this? Where will I put my 5 prime? See here, this is 5 prime, this is 3 prime. So where will I put my 5 prime? Here, right?
Yeah.
5 prime to what?
3
Prime. So can you see that the replication is running in opposite direction?
Yeah.
Yes. Yes or no?
Yeah.
Isn't the replication running in opposite direction?
Yeah.
Yes. Take a minute and see. Try to understand. If you have any doubt, clarify it. Don't pile up your doubts. Others later you'll not understand. Clear? Anything?
Understood. Yeah.
Clear? Okay.
Yeah.
So 5 prime to 3 prime. This region is called as replicating fork. Okay. So here, what we will do is that we will now add, okay, here, just where the 5 prime is, we will first add one enzyme, or we will add one nucleotide. This is called as primer. What is it called? Primer. It is an RNA. So this RNA will now add one nucleotide. Okay? one nucleotide. It will go on adding nucleotide. One nucleotide will go on adding and it will make one strand. But since the replication is in the opposite direction, away from the replication fork, see here, this in this strand, the replication is away from the replication fork. So the RNA primer is not able to continuously add in the nucleotides. So again, it will add one primer. Then this primer will add again the nucleotides for some distance. Again, we need one primer. Okay, again, then only it will add the nucleotides. Now, what do you understand by nucleotides? Suppose, Hannah, this is the template strand. In the template strand, we have adenine, guanine, cytosine, thymine. And we are forming the new strand. So can you tell me what will be here?
T.
Thymine. Okay, here.
Cytosine.
Yes, here.
Guanine.
Guanine. Yeah, here.
And
Yes, adenine.
adenine.
See, aren't they complementary?
Yeah.
Matching?
Yeah.
Yes, we call them as complementary base pairs. So that's how they will read the complementary base pair. If here there is an adenine, primer will bring a thymine here. Are you understanding?
Yeah.
Yes. So like this, they will make the nucleotides. But what is here? Is the replication continuous? Is it continuous replication?
No.
No. So in this strand, the replication is discontinuous. So it is called as lagging strand. Clear?
Yeah.
Okay. But see here. But what happens here? Only one primer will be there. This replication, can you see it is towards the replicating fork?
Mm-hmm.
Here, the replication is towards the replicating fork. So here, the nucleotides will be continuously added. Continuous addition of nucleotides. Okay? So here in the strand, replication is continuous. So it is called as the leading strand. This point is clear?
Yeah.
Yeah. Now, who will decide if it is adenine, thymine to add, guanine, cytosine? So after all this, we have helicase, single-stranded binding protein. We have here a major, I'll write it here. We have here a major enzyme which will add the nucleotides. And this enzyme is none other than DNA polymerase 3, one of the most important enzyme of replication. This is the one that will control every part of replication.
Mm-hmm.
Is this point clear? Yes, clear or not, Sana?
Yeah.
Yeah. Understanding anything you are doubtful, have a look at the picture. When is your test?
On Tuesday and Wednesday. So
Okay.
Tuesday is paper one.
Sorry?
Tuesday is paper one and Wednesday is paper two.
Okay. This is clear?
Yeah.
Okay. Now see. All right. Now you see here. I'll just draw this. Okay. See. What did we have here? We had here 3 prime. This is 5 prime. Here this is 5 prime, 3 prime. So what we had here? Here discontinuous replication, right? And here continuous replication.
Yeah.
So yeah. So this is one discontinuous and continuous. I'm not drawing the enzymes. Okay. Now, when the replication is completed, when the enough DNA is, whatever is the required DNA that is produced, that is completion of replication. Clear?
Mm-hmm.
Okay. Now, let's see here. Let's see here. So, we cannot have a DNA with the primer. So, we have an enzyme which is called as DNA polymerase 1. This DNA polymerase 1 will go and remove all the primer. DNA polymerase 1 will remove primer.
Mm-hmm.
Clear?
Yeah.
Now, when DNA polymerase 1 removes the primer, DNA polymerase 1 will remove the primer. Okay? So, we will have like this here and this here. There are gaps created, right? See this?
Yeah.
When we remove the primer, there's a gaps created. These gaps. This is called, okay. So these gaps are created. So when you see this DNA, don't you feel that the DNA is in fragment? Is it whole DNA?
No.
No, it is in what? Pieces, right?
Yeah.
Can we have DNA which is in pieces?
No.
No, we should have an entire DNA. Okay, we should have an entire DNA. So here, these fragments are called as Okazaki fragments.
Mm-hmm.
Okay, what are they called?
Okazaki fragments.
Hmm. Okazaki fragments. Clear?
Yeah.
Yeah. Okay. Now, next. So now DNA, again same, DNA polymerase 1 will now, what they will do? They will now add, they will fill the blanks, fill in the blanks, like we do know, fill in the blanks. They will fill the gap. But see here, a small gaps are still there, right?
Yeah.
So this is not full DNA. So we will have the enzyme ligase that will seal the gap. Once the ligase seals the gap, we get a complete set of DNA. See?
Mm-hmm.
Okay. Now this section, there will be a cut in this section and the two new DNA will be separated. This point is clear?
Yeah.
And this marks your replication.
Mm-hmm.
Termination of replication. Replication is over. Okay, so DNA polymerase has two functions. It will remove the primer and it will add the nucleotides to complete the Okazaki fragments. Clear?
Yeah.
Okay, yes. So this is what a replication is. All right, Sana? Yes?
Yeah.
Yeah. And one more thing, Sana, once you're done with the, your exams, okay, you can just send over your test paper. I want to see the, how you have written the test paper. Okay. Can you send over the questions? Yes.
Yeah, I will.
Okay, both paper 1 and paper 2. Sent over the questions. Okay, if you have not understood any question, maybe I can clarify that. Okay, but for after every test, please send over the questions. Okay, so I can
Yeah.
just check out. Okay, clear? Okay, this is clear, Sana?
Yeah.
Yes. Okay, so this is a replication. Now, small few important things that you need to know. Okay, that you need to know is carbohydrates, proteins, lipids. Carbohydrates, proteins, lipids. Okay, Sana, which is your favorite food?
Probably,
Sorry?
I think pasta.
Pasta. Okay. All right. Pasta. Okay, fine. Okay. What does pasta contain?
Carbs.
Spaghetti, pasta. Yes. Spaghettis, pastas, aren't they made up of carbohydrate?
Yeah.
Yes. Carbohydrate. That is starch, right?
Hmm.
Yes. Okay. Whatever the pasta that you're taking in, okay, the pasta that you're taking in will contain what? The spaghettis. Okay, the pasta which is made up of flour, which is starch,
Hmm.
right? Okay. The pasta will be fried, or sorry, which will be prepared in olive oil. Yes,
Yeah.
a little bit of olive oil will be there. Yes,
Yeah.
oil is lipids.
Hmm.
Okay, and then if you add chicken in the pasta, you have innovations. You have you're taking in the proteins. Carbohydrates, lipids, and proteins. They are the three classes of food that we take in every day. Okay, every day. Are you feeling sleepy?
No.
Okay, we take in every day. Carbohydrates, proteins, and lipids are the food that we take in every day. So
Hmm.
we need to know what is carbohydrate first of all. Okay, so let's see here. Carbohydrate. Carbohydrate. Carbohydrates. These are sugar. They are called as saccharon. What they're called? Saccharon or saccharides.
Hmm.
Saccharon or saccharides. Carbohydrates. They are the important source of energy in our body. Carbohydrates. They are classified. They are classified into three categories: monosaccharide, oligosaccharide, and polysaccharide. Monosaccharide, these are simple sugar. They cannot be further hydrolyzed or cannot be further broken down. Broken down. One more thing. Carbohydrates are made up of compounds of carbon, hydrogen, and oxygen. Carbon, hydrogen, and oxygen. This point is clear?
Yeah.
Yes. Okay. All right. Let's see here now. Monosaccharides, I said they are the simplest sugar. They cannot be further hydrolyzed. They cannot be further broken down. So we have some of the monosaccharides. Okay, monosaccharides like glucose. Glucose is an important monosaccharide. Okay, glucose is an important monosaccharide which is present in almost all and very common. It's a common sugar, simple sugar. Okay, so glucose structure is C6H12O6. It has 6 carbon atom. It has 12 hydrogen atom and 6 oxygen. So the structure you need to know. Clear?
Yeah.
Structure is important. Glucose. Glucose is called as blood sugar. It is present in our blood. Glucose is present in, okay, it is present in cane sugar. Okay, it is present in cane sugar. Cane sugar, sugar cane, right? Okay,
Yes.
along with fructose. All right, so cane sugar is called as sucrose. So sucrose contains glucose plus fructose. Fructose is the sweetest sugar. It is a fruit sugar. It is present in all fruits except grapes will have glucose. So glucose is also called as grape sugar. Remaining all fruits will have fructose. Grape will have glucose. Okay, we find glucose and fructose are the two important monosaccharide. We find glucose in honey. Honey, we will find glucose and fructose is found. Okay, monosaccharides, they cannot be further hydrolyzed. Glucose, when we eat the pasta, okay, when you eat the pasta is digested in your digestive system. The glucose, the starch is broken down into glucose, and this glucose will be absorbed, which will give you energy. So glucose, it is from glucose that we get energy. Glucose is broken down in our cells. Okay, we call it as, see, metabolism of glucose is called as glycolysis. Glycolysis will help us to produce ATP or energy in our body. How will you define metabolism? It is the sum total of all chemical reactions in our body. Whatever chemical reactions are occurring in our body, that is called as metabolism. Sum total of all chemical reaction in our body is called as metabolism. Clear?
Yeah.
After oligosaccharides will contain 2 to 9 monosaccharide units, okay, joined. So there will be many monosaccharides will be there. 2 to 9. How many? 2 to 9 monosaccharides. When they are joined by what bond? They are joined by a bond which is called as glycosidic bond. And when they join 2 to 9 monosaccharides unit, when they are joined, they'll form oligosaccharide. And the most important oligosaccharide we study about the disaccharides. Di meaning two monosaccharide unit joined to form a disaccharide. Like cane sugar. What is there in, who's there in cane sugar? Yes, Sana, who's there in cane sugar? Which monosaccharide?
Start. Glucose.
Glucose and fructose, right? See? Yes or no?
Yeah.
Yeah. Okay, cane sugar, glucose and fructose. Then we will have lactose. Lactose is present in milk. So we will have glucose plus galactose. Okay, glucose plus galactose. Next, maltose, malt sugar, will have glucose plus glucose. When two monosaccharide unit joined, okay, they form the disaccharide. This point is clear, Sana?
Yeah.
Yes, glycosidic linkages are there, right?
Yeah.
And when we come to polysaccharide, polysaccharide are called as complex carbohydrate. They have 10, they have more than 9 monosaccharides. Thousands and thousands of monosaccharides will join by glycosidic bond to form polysaccharides. Many monosaccharide units, many monosaccharide, okay, they join to form what? Polysaccharide. Polysaccharides are complex carbohydrate. One of the polysaccharide that you ate, pasta, contains what? Starch.
Mhm.
Starch is made up of many glucose molecule. Many glucose what? Molecule. Clear?
Yeah.
Okay. Next. Starch. Glycogen. Starch. It is the storage product in plants. The carbohydrate is stored in the form of starch in plants only. Glycogen is the carbohydrate which is stored in animals. Okay, we store glycogen in our liver and also in the muscle. Glycogen is made up of, again, many glucose molecule. Many glucose molecule. Clear?
Yeah.
Yes.
Yeah.
Then cellulose. Cellulose. Okay, cellulose. This is also a polysaccharide which is made up of what? Glucose. Okay, cellulose is found in the cell wall of plant cell. These are fibers. We humans do not have enzymes to digest polysaccharide, that is cellulose. We don't have enzymes to digest cellulose. This point is clear, Sana?
Yeah.
Yes. Okay. Carbohydrates. Then lipids are fats. Okay, lipids will be fats, and lipids are made up of fatty acid plus glycerol. All the fats that are present in the body, they are stored in a particular cell. Okay, this cell is called as adipocyte. This is the fat vacuole. Adipocyte. Okay, adipocyte. Where is the adipocyte present? It occurs as a layer, and we call this layer as what tissue? Adipose tissue. Okay, adipose tissue. It's found below the skin. It is present below the skin. We have cholesterol. We have BVAX. All are lipid compound. Okay, all are lipid compound. Lipids, when we eat, they are digested. Okay, lipids, when we eat, they are digested with the help of lipid digesting enzyme or lipases. Okay, the lipids molecules, they are joined by ester linkages. Carbohydrate will be joined by glycosidic bond. Lipids are joined by ester linkages. If we see protein, proteins will contain many amino acid joined together. Okay, this is one single amino acid. They are joined together by peptide bond to form what? Protein. So protein is called as polypeptide chain.
Mhm.
Okay, it is called as what? Polypeptide chain. Amino acids. If you look into the structure of the amino acid, we have COOH group, NH2, hydrogen, and R. This is the variable group. Many other groups can attach to this R group and produce different type of amino acid. For example, you can add hydrogen here, or you can add hydroxyl or an aromatic compound. Depending on what group is added to that R group, that will determine what amino acid it is. And all of this is the amino group. We call it as the N-terminal end. This is the carboxylic group. We call it as the C-terminal. Everything is attached to the central carbon. We call it as the alpha carbon. Alpha carbon. Okay, we call it as the alpha carbon. All right. So this is one single amino acid. Like this, many amino acids will be joined to form a protein. Okay, to form what? Protein. So amino acids or proteins contain nitrogen, carbon, oxygen, hydrogen. Okay, these all will be present in proteins. Now, we have 20 different type of amino acids, and this 20 different type of amino acids combine in different manner to make many proteins. Very important. Many proteins in our body. Is this clear?
Yeah.
Yes. Okay. So we'll have peptide linkages. I told you. Okay. Peptide linkages. These are linkages which are present between the these are linkages which is present between the two amino acids. If this is, if you remember, one second. See, this is the one that we had done last. See, in the previous class.
Yeah.
You can see this. Do you remember this?
Yeah, I do.
Yes, this is I have told you about the formation of what bond? Peptide bond. See, this is the formation of the peptide bond. See, this is a peptide bond. Can you see this? Right? This is a peptide bond. So refer to this previous notes also that I had given. Okay, the previous revision class. I've said you that you refer to that notes as well.
Yeah.
All right, refer to that notes as well. Okay, enzymes. What are enzymes? What are enzymes? If I define an enzyme, enzyme is referred to as a biocatalyst. Okay, they accelerate the rate of a reaction. Any reaction, they will accelerate. Accelerate meaning the reaction will occur faster. Not only that, not only the reaction will occur fast, but we will also get more number of products. Clear? We'll get more number of products. So suppose, for example, if this is the substrate, this is the substrate, and we have here, the enzyme. So this substrate will bind in this part of the enzyme. So this site where this is the enzyme, this site where the substrate, what is it called? Can you tell me? What is this site called? active site, right? Now this enzyme binds to this active site and produces numerous, it will break the bond. It will make the bond. It's like lock and key. We have many substrates. Every substrate will not fit in every enzyme. Okay? Only specific substrate fixes in specific enzyme. So this theory is referred to as the lock and key theory. The enzyme will bind and what it will do, break and make bonds. After that, okay, what it will do, it will make out of the substrate, it will make a new product. Clear?
Yeah.
Different type of enzymes. Just now we studied in DNA replication. What did helicase do? Helicase broke the bonds. What did ligase do? Ligase joined the bonds, right? So different enzymes perform different functions. Clear on this?
Yeah.
Yes? Okay, so just go through the previous notes that I had given. All right, so I hope the molecular biology, most of the things are clear now, Sana. Yes?
Yeah.
Yes. Okay. All right. So do one thing. Just immediately send me the picture, okay, of the textbook.
Is it possible if you could send me like past questions about molecular biology?
Past questions. Yes, I have the past questions. Okay, so I have a class. After the class, I will send you.
Okay.
Okay. All right. After the class, I'll send you. And I'll do one thing. These are past question papers. Okay. So I'll just send you the question papers. And these question papers may also have questions from other chapters as well. So you can keep them as a reference, right?
All right.
And we can use it. Clear? All right. Okay. That will be fine. Okay. And some other questions also I'll send you. Then, yes, you need to send me the picture of the book.
Yeah.
Okay. All right, just immediately after the class, take a picture, scan it and send it. Okay. And second thing, for every test, send me your question papers. I want to see, okay, how you're writing, and I'll be asking you how did you write and all. Okay. All right, so don't forget. Okay, chalo, bye. Take care. All the best for your exams.
Thank you.
Okay, bye. Take care.